all the useful functions of antibody depend on its ability to combine with the corresponding antigen to form an immune complex. Hence, immune complexes, also called antigen-antibody complexes, are the lattices of antigen and antibody. They can either be localized to the site of antigen production or may circulate in the blood. Now, these antigens can be derived from an infection, innocuous environmental substances, or it can be an autoantigen. Immune complexes are produced as part of the normal immune response. But do you have any idea how does their clearance occur? Well, the normal fate of these complexes is phagocytosis, which is greatly enhanced if complement becomes attached to the complex. However, there are circumstances when this fails to happen, particularly if the complexes are small, where more than one antigen is bound to each antibody. This usually happens when there is an excess of antigen. Like in persistent infections and in autoimmunity, where the antibody is of very low affinity, or where there are defects of the phagocytic or the complement systems. If not rapidly phagocytosed, complexes can induce serious inflammatory changes in either the tissues or in the walls of small blood vessels, depending on the site of formation, and immune complex diseases may arise. So, this entire video revolves around these three things I just mentioned: immune complex, complement, and immune complex diseases. Immune complex diseases are the third form of hypersensitivity in the Gell and Coomb classification, called Type III Hypersensitivity. Well, hypersensitivity is an exaggerated or inappropriate immunologic response occurring in response to an antigen or allergen. Now, these hypersensitivity reactions are classified into 4 types according to Gell and Coomb's classification. Type 1, called Anaphylactic or immediate hypersensitivity reactions. Type 2, called Cytotoxic or antibody-mediated hypersensitivity reactions. Type 3, or immune complex-mediated hypersensitivity reactions. And Type 4, known as delayed or T-cell mediated hypersensitivity reactions. You can memorize these types easily by the mnemonic acid, where A is for anaphylactic, C is for cytotoxic, I for immune complex, and D for delayed. Our topic of concern for today is Type 3, the immune complex-mediated hypersensitivity reactions. Well, Type 3, easy to remember, because 3 things are stuck together here: the antigen, antibody, and the complement. In Type 3 hypersensitivity reactions, IgG antibodies bind to circulating antigens to form an antigen-antibody complex. This complex then deposits in particular tissues, and those tissues are mostly blood vessels, the basement membrane of lungs, kidney, and in the joints. This deposition activates the complement cascade and attracts neutrophils. Now, what do neutrophils do? Yes, neutrophils will release lysosomal enzymes and cause cell death and inflammation. Few examples of Type 3 hypersensitivity reactions or immune complex diseases can be memorized by the mnemonic SPAR, where S stands for serum sickness and systemic lupus erythematosus, or SLE, P for polyarteritis nodosa, and post-streptococcal glomerulonephritis, A for Arthus reaction, and R for rheumatoid arthritis. Now that you have got an idea of the topic, what is hypersensitivity? What are the 4 types of hypersensitivity according to Gell and Coomb's classification? What is Type 3 hypersensitivity, and what conditions are included in it? Now, I will be discussing the immune complex, with a special focus on the types of antigens in the immune complex, antibodies involved, the clearance mechanism by complement, and lastly, the immune complex diseases in detail. So, do not forget to click the next part.